Please tell me the number of power. I must start from this section for teaching. I don't remember the number of power. Silver composition. Thank you very much, Mr. Mahavi, Qasim Mahavi. Okay, so we finished. I think we finished power 2, yes? We must start from power number 3. Yes, I upload the wrong. This is power number 2. I'm going to upload the power number 3. Yes, I'm going to upload another game. Okay, now we are in power number 3, numerical taxonomy. We are getting the number 3 is get started from here, and we must study this cell wall composition. Okay, thank you. The polypeptide, this type of categories are in phenotypic group that I told you the previous section. The peptidoglycan component of cell walls of bacteria does not provide much information except for classifying into gram-positive, gram-negative, and acid-fast bacterial types. So the peptidoglycan composition is very, very important, and for classifying to gram-positive and gram-negative is used, and acid-fast bacterial types also. How, but not more, okay? Just for classifying the peptidoglycan. However, those in gram-positive cells contain different types of peptidoglycan depending on the genus or species. The peptidoglycan structure can be analyzed by determining its type. So, in gram-positive cells, we can see types of peptidoglycans depend on their different kind of peptidoglycans, depend on their species, and the peptidoglycan structure can be analyzed by determining its type. So the cell wall composition can be defined in bacterial cell, and the most of this composition is related to peptidoglycan composition. So we can determine the component of peptidoglycan in gram positive, gram negative, and also acid fast bacterial cells. A or B mode of cross-linking, whether it's directly linked or via interpeptide bridge, and with amino acids in the bridge, and the composition of amino acids, especially the diamino acid of the side chain. So we have cross-linking inside the peptides, and we can see in the structure of peptidoglycan some cross-linking, as I told you before, and we have interpeptide bridge too with amino acids in bridge. We have different amino acid bridges, so we have different peptidoglycans, and the variation of peptidoglycan is due to this variation of amino acids that are specially specific in each species. While the mode of cross-linkage can vary between species and also between strains, the amino acid composition is common to all species within a genus. So you can see, while the variation of cross-linkage and interpeptide bridge amino acids is observed, and the amino acid composition is very different from one species to another species, and also between strains. That is very important. And also between the strains we can see this variation. In archaea, sodomurane is present where N-acetyl muramic acid is placed by N-acetyl talozuronic acid. So here you can see another variation between bacteria and archaea. In archaea, sodomurane. In archaea, we have sodomurane that is present where N-acetylmuramic acid is replaced by N-acetyltalozuronic acid. So in archaea, we have N-acetyltalozuronic acid instead of acetylmuramic acid in bacteria. So here you can see another variation in the peptidoglycan structure. So the amino acids are different from one species to another, one strain to another, and also in bacteria and archaea we can observe differences between amino acid compositions, between peptidoglycan compositions. Okay, another phenotypic taxonomy is approach about is fatty acid analysis. You studied, if you remember, you studied about polyphasic taxonomy, the previous section, that is divided to genotype and phenotype methods. Now we are studying phenotyping methods. So fatty acid analysis is a method for taxonomy of bacteria relate to phenotypic characteristics. Different types of lipids are present in bacterial cells. Polar lipids are present in the lipid bilayer of the cytoplasmic membrane. The diversity of polar lipids is known to be large and many are yet to be structurally elucidated. So, the main structure of the lipid in bacteria are polar lipids. The structure of fatty acid is contained with polar lipids, and the diversity is observed in fishes. While in archaea, polar lipids are types of phospholipids, aminophospholipid, glycolipids, and phosphoglycolipids in bacteria. Apart from the ones seen in archaea, there are also lipids derived from amino acids, capenins, sphingolipids, glycol or phosphosphingolipids, and hopanoids. Okay, so uh, as I told you, in bacterial cells we have polar lipids, we have phospholipids, but in archaea we have some amino acids, some capenins, sphingolipids, glycol, or phosphosphingolipids are the most important structure of lipids in archaea, and hopanoids also. And hopanoids it can be observed in bacteria. In archaea, we have polar lipids, we have phospholipids, aminophospholipids, and glycolipids as and phosphoglycolipids. So in bacteria, we have phospholipids. In bacteria and archaea, this type of fatty acid is common, but we can what we see a variation between and some differences between bacterial fatty acids and archaeal fatty acids. So you can see the type of different fatty acids here in bacterial cells and archaeal cells. And the polar lipids in bacterial cell are contained from phospholipids. And what are the hopanoids? Hopanoids molecule, uh, they are exist in bacterial cells and they are like estrols. You know what the estrols kind of lipids that we can observe in Mycoplasma genus. Mycoplasma don't have any cell wall, and they have estrol in their cytoplasmic membrane. Why? Why they have estrol in their cytoplasmic membrane? Do you know? Here it will be clear how much of the students are present in class. So my question was that why estrol is exists in mycoplasmic membrane, you know? If you're really in the class, you must answer. Okay, I will tell you, but I think from this class that 25% are present with their names, but really only 3 or 4 of them are present in class. Ali Hamid, Haydar Qasim Mahavi, and Ruya Ehsan Ahmad. They answered me, so they are only the present in the class. But others are doing something else. I don't know they are where, but just their names are present here, fortunately. So I'm going to tell you the yes, I'm going to write, but if they listen to me, they understand. Why? I'm typing for you. Why esters are present in mycoplasma membrane. Why esters are present in mycoplasma membrane? You know, mycoplasma is the genus of bacteria that they don't have cell wall, okay? But their lipid is strong in cytoplasmic membrane. Why we have estrons only in mycoplasma genus? Okay, Mr. Ali Hamid, because lipids are a component of the plasma membrane. Yes, lipids are, but esters are a kind of lipids are only exist in mycoplasmic membrane. I'm going to tell, and I think only you are present in the class. Only three of you, all of the class. Mr. Qasem Mahawi, Miss Roya Ihsan Ahmad, and Mr. Marve Ali Hamid. Only three of students are answering my question. I'm going to tell you, the straws are only present in the microplasmic membrane. Why? Because they don't have any cell wall. Okay, the mycoplasma genius in bacteria don't have any cell wall. So these kind of lipid, I mean straws, can uh, protect the cell instead of cell wall. Okay, a cell wall, and these straws can give rigidity to the cell cytoplasmic membrane. This was yes, Mr. Ali Taleb, but the answer for my question is that the rigidity instead of cell wall can be provided with esters. Okay, now we are going to continue. So, hopanoid, I was telling hopanoid. Hopanoid are very similar to esters. They give rigidity to the plastic membrane. They can shape and they can do many actions in bacterial cells, a kind of lipids, very similar to esters. In gram-negative bacteria, lipopolysaccharides are present in the outer membranes, the type of sugar present and the fatty acid type. The linkage of the fatty acid to the sugar amide or ester linkage provides information on characteristics of the cell. However, determination of lipopolysaccharide is not routinely used in recent times. So in gram-negative bacteria, we have lipopolysaccharides, you know, that is very special for these cells. And the type of sugar and fatty acid is a little different. So when we investigate, when we approach about the um, lipopolysaccharides and fatty acids in gram-negative bacteria, we can distinguish them and we can characterize. Determination of lipopolysaccharides is not routinely used in recent times, but this was used in the past. Okay, if you don't have any question, you can ask me. Okay, so we continue the lecture. The total cellular fatty acids are extracted, esterified, and the methyl ester content is analyzed by gas chromatography. Under standard conditions, this provides a reliable estimate of taxonomy up to genus and sometimes a special level. I think I have sent this power for you, haven't I? I think I have sent it. Am I right? You must have this power number 3, taxonomy number 3, I think. Okay, so the total fatty acids that we can extract from cells can be different. For example, methyl ester content can be recognized by gas chromatography. We are talking about analyzing fatty acids. So methyl ester content is analyzed by gas chromatography. Under standard conditions, this provides a reliable estimate of taxonomy up to genus and sometimes species levels. So under standard conditions, this is very important to estimate of taxonomy for genus and sometimes species levels. So please note that under standard condition, uh, this is very variable between taxonomy and genus. For example, uh, between different genus and different species that are different. Okay, the next phenotypic characteristics that we are going to study is a Plano Equinox. As a phenotypic taxonomy, we can use isoprenoid quinones approach. So we can characterize isoprenoid quinones that are very important for the taxonomy of special bacteria. The respiratory isoprenoid quinones are components that occur in cytoplasmic membranes of prokaryotes, archaeas bacteria. So we can see isoprenoid quinones in the both archaea and bacteria together. So we can see the quinones in bacteria and archaea together. The naphthoquinones with subtypes quinone and benzoquinones, rhodoquinones, and plastoquinones are the major types of quinones. Please note that the major types of quinones are numbered here. I read it one more time. Naphthoquinones, that is divided to phylloquinone and menaquinone. Benzoquinone is divided to ubiquinone and rhodoquinone and plastoquinone are the major types of quinones. Okay, isoprenoid ether-linked side chains are present in membrane in members of the archaea. They are of different types such as diethers, hydroxylated ether diethers, macrocyclic diethers, tetraethers, and polyol derivatives of the tetraether. So isoprenoid. Ether-linked side chains are present in many bacteria, as I told you, and different type of dieters we can observe in the bacterial cells that are very important. Why? Because when we detect these isoprenoid, these ether-linked side chains, we can guess that we detect the archaea cell. So there are different types of dieters, such as hydroxylated ethers that we can see in archaea, macrocyclic dieters, tetraethers that are very common in bacterial in archaeal cells. They are not of bacteria. Please note, they are only exist in archaeal cell. We can observe these type of components only in archaeal cells, so we can distinguish that is archaea. Tetraethers and polyol derivatives of tetraethers. Okay, non-isoprenoid-based ether-linked lipids are present in bacteria and can be straight chain or with sample branch side chains or saturated derivatives. We have, okay, do you have my sound right now? Mr. Qasim Mahawi or Mr. Jaber Amit, Marve Ali Hamid, or Roya, Ehsan Ahmad. The problem is for um, my voice is clear, but you cannot hear. I think these students were not present really in the class, and this is uh, I don't know what. I think no sound or no hearing sound is just explanation for not presenting in the class. I think not presence or not operation in class. Not, I don't know. Okay, non-isoprenoid. Okay, thank you, Mr. Vassam Jobber Abid. Thank you for your explanation about my sound. Okay, now non-isoprenoid-based ether-linked lipids are present in bacteria and can be straight chain or branch side chains or with monounsaturated derivatives. So we can see the variation here. We can see base ether-linked lipids in bacteria, and we can see simple branch side chains, and we can see monounsaturated derivatives that are observed in bacterial cells. And non-isoprenoid base ether links are present bacteria. Please notice isoprenoid ether link sites are observed only in the archaeal cell. Isoprenoid are only in archaeal cell, are not in bacterial cell. So non-isoprenoid based ether link lipids are present in bacterial cells. And isoprenoid is, uh, you know, it is a solicitation for some components in bacterial cells. So isoprenoids are observed in archaea and non-isoprenoid lipids are observed. So isopronate is instead of fatty acids in archaeal cells. Uh, some of the students have my voice very clearly, so I think this is because of your net or the internet, I don't know. Okay, and now we are going to study about other diagnostic methods. Sorry, if you have any question, you can ask me. Other than the principal diagnostic methods described above, other techniques used to lesser levels or for comparison between species or strains comprise of the following. A. Whole cell protein analysis, wherein protein is extracted from the cells and analyzed by sodium diacetyl sulfate polyacrylamide gel or STS page, gel electrophoresis. So here you can see other diagnostic methods. For example, whole cell protein analysis. We can analyze the whole cell protein and then we can diagnose the bacterial cells. Or when we want to extract and then we want to realize the proteins and we want to extract the proteins, we must use sodium diacetyl sulfate polyacrylamide gel electrophoresis or SDS page. This is an appropriate method for analyzing the cell. So the first method for diagnosing the bacterial cells are contained cell protein analysis. This can help in comparison between related. The next method is polyamines that are a group of compounds in the cytoplasmic that provide stability to the DNA and maintain osmolarity in the cell. So polyamines are a group of compounds in the cytoplasm that provide stability to the DNA and maintain osmolarity in the cell. They are useful to distinguish above genus level and level 2. So you know polyamines kind of molecules that they can provide a stability to the DNA. Because why? Why do you know why? Because they have positive polarity and they can neutralize
شكرا.
the negative polarity of the DNA. So they can provide stability to the DNA molecules. Polyamines with positive charge and DNA with negative polarity. So they can neutralize and they can provide stability to the DNA molecule. We can recognize this kind of molecules, polyamines, for dividing for taxonomy, and they are useful to distinguish the genus levels and also species levels, up to species levels too. The third kind of the molecules or cytochromes are associated with the cytoplasmic membrane and are involved in respiratory and phosphosynthetic electron transfer. So cytochromes are important in cytoplasmic membrane and are involved in respiratory and photosynthetic electron transfer. They are important molecules, you know. We can realize, we can analyze these molecules, we can distinguish them, and then, for example, we can um, recognize the photosynthetic bacteria. Okay, we was reading about phenotypic and polyphasic taxonomy and advanced spectroscopy and spectrometric methods are another techniques for phenotypic methods. Many advanced analytical techniques such as pyrolysis, mass spectrometry, Fourier transform, infrared spectroscopy, and UV resonance, Raman spectroscopy have been employed to determine chemical composition of bacterial cells. So we have many advanced analytical techniques such as mass spectrometry, infrared spectrometry, and UV resonance Raman spectrometry that they are a very good, very good methods, and we name them advanced spectrometry. Advanced spectrometry for determining the composition of bacterial cells, so we can determine the composition of bacterial cells, and we can distinguish species. Okay, and the next title is genotypic technique that I'm not going to teach you anymore. I told you because your uh, lessons are not behind and the volume of your lecture um, will be enhanced. So I stopped teaching until the genotypic techniques, and I will start from here for the next section. Please write the number of power number 13, genotypic techniques that we are going to start next week. So please write the number of 13, Qasim, Mr. Qasim Mahabi. Number 13, thank you. Okay, thank you. I'm I'm shortening the lecture. I teaching 10, 12 slides each section, and that is a good volume of explanation or texts for your teaching. It's not too large and too long. I can teach you for 80 minutes, but I'm teaching you for 60 minutes and up to 10 or 12 power. That reason is be shortening the volume of your lessons. And that is why I'm doing it. Okay, yes, I know, Mr. Imam Muhammad Ali. Most of you, unfortunately, are doing your job during official work. I'm just talking about checking for two or three or four of students, but another students are doing something else. I don't know. I know. Unfortunately, uh, the duration and the period of this lecture is very, very short, and uh, I will be happy to be present in that. Okay, so your lecture until uh, genotypic techniques, and I say goodbye until the next week. Be healthy. I hope for you a nice week. Take care of yourselves. Goodbye.